Good morning, sir.
Good morning. Shall I start now?
Good morning, participants. Yes, sir. Good morning, participants. Today we have again our professor, resource person, Professor Tapan Kumar Adhyay, sir. He is director from South Asia Nitrogen Center and professor in School of Biotechnology, Kalinga Institute of Industrial Technology, Bhubaneswar, Odisha. I welcome sir on behalf of the participants and Department of Botany, Telangana University for the lecture, sir.
Yep. So I am starting then, okay?
Yes, sir.
Good morning, participants. Today I'll be speaking about the sustainable agriculture and the role of microorganism in the process. So this is actually, over the almost 5,000 years of the human history of habitation, where the people start dwelling together in a society. Before that, they were mostly hunter-gatherer. That means for their food requirement, they used to either gather from the wildly growing plants or trees, fruits, etc. You can remember, imagine the picture of Mowgli going and picking up the banana from the banana plant, almost like that. It was a hunter-gatherer and they kill the animals if they require some meat. So that was the habit of the people. And when they started gathering together in the form of a society, then they found that the resources are not enough. They have to grow their food. And that's how they started agricultural operations, I would say, very rudimentary. And just a minute. Yeah, this is a picture from the Egyptian mummy houses, where it is shown that some person is trying to do some cultivation. They are tilling the soil with the help of animal power and trying to grow the foods which you can see at the bottom, and the crops that are grown here. And this is how people started agriculture, and then slowly it developed under specific scenarios, like mostly in the river banks or the riverine areas where plenty of water supply was there. And you had all those civilizations like Mohenjo-daro, Harappa, Indus Valley civilization, or even for any other civilization. They're mostly near the riverine areas. And during monsoon, they used to overflow. There used to be flood, and that also carries nutrients in the form of silt and deposit over the soil, and gives a bountiful crop for their requirement. Limited number of people. So obviously that was that is how the agriculture started. Now, let us come back to the present day scenario. The United Nations estimates world population will be around 9.8 billion people by 2050. We are now around 6.8 or something like that. And it will be around 9.8 billion by 2050. Now, in order to feed them, the agricultural productivity must increase. Of course, the farmers, they have done their job. Scientists or the research scientists, agricultural scientists, they have also done their job in providing resources, some resources, and the farmers, they did their job in producing, so that the population has increased to a certain extent. But with the continuity of the population growth, it is expected to touch 9.8 billion people. And in order to feed them, there is to be an estimated 70% increase for the demand for food, feed, fiber, and bioenergy. So this has to increase, and because arable acreage is unlikely to grow, land is becoming limited day by day because population has increased and there has been so many ancillary activities, industry, road, building and all. So now land is not available. Now under these circumstances, what we call as the growth of the agriculture has to be vertical. That means it has to grow in a smaller area through more intensive agricultural practices. Earlier it was horizontal. That means it's spread on new and new land area available. But then now, since the land area is restricted, so the arable acreage is unlikely to grow. So meeting this demand requires achieving higher yields through application of artificial fertilizers and pesticides, now whose manufacture and uses are not suitable. Now, if you think of the fertilizers and different synthetic fertilizers, their production is energy intensive. You know that all this nitrogen fertilizer, urea, what we farmers use in the field, they are produced in factories which requires a lot of energy, either in the form of natural gas or mostly fossil fuel, then phosphorus and potassium. Now this while nitrogen we don't have any problem of their resource. I mean, it is nitrogen is everywhere, and 78% is of the atmosphere is nitrogen. But for phosphorus and potassium, we have to mine from the finite sources. And there are estimates that probably phosphorus and potassium at the current rate of consumption may be depleted within 100 years. These are the projections, of course. Now, pesticides with carcinogenic, developmental and environmental risks are restricted. We know now because these are anthropogenically developed chemicals. These are not normal natural chemicals. These are all artificially synthesized. They have got their impact on the health, and some of them are carcinogenic. They have got environmental risk. Now, more sustainable strategies to achieve this very higher crop yield under this scenario. Now I am showing this picture. It's actually two pyramid pictures, and then one is for the food pyramid, another is the environmental pyramid. This I wanted to show that production of food has impact on environment, and interestingly, some of the easily available things as food has got low environmental impact. And more we complicate it, our environmental impact goes on increasing. Okay. Now, say for example, the different and their production also will be little bit less. Now, if you see the food pyramid, now the different grains and etc., they have got a lower environmental footprint, and then once you go on increasing, go on different types of refined food and modern these things, you see that they have got a high impact on the environmental scenario. Like if you take different things like fruits and etc., they have got a lesser environmental footprint. But when you go up with meat or beef or chicken and all, they have got a higher environmental pyramid. So this I just wanted to tell you that our food habit also determines our environmental footprint. Now, this has developed concern among the policymakers that how we can reduce the footprint. It is not possible that we stop eating and then there will be no footprint, environmental footprint. We have to eat. And for eating, we have to produce the food. And that will also have its own environmental impact, as I said, using fertilizer, pesticide, etc. So the concept came of conservation agriculture. This is a concept for resource saving agricultural crop production. You save your resource, but at the same time continue to produce your agricultural production that strives to achieve acceptable profits together with high and sustained production levels while concurrently conserving the environment. This is a FAO definition of 2007. So some of the things that is coming here from this definition is that it's a resource saving agricultural crop production, and this is strives to achieve acceptable profit, strives to achieve acceptable profits, and with high and sustained production levels. That means initially when the conservation agriculture concept was developed, it was thought that okay, whatever you produce, go on producing that much only, so that system environment is conserved. But it's not like that. You continue to have high and sustained production level, but concurrently conserving the environment. So this was the conservation agriculture. Now, there are three basic principles of conservation agriculture. One is practicing minimum soil disturbance, which is essential to maintaining minerals within the soil, stopping erosion, and preventing water loss from occurring within the soil. Now, this is a very interesting thing, because when the modern plowing equipment, agricultural equipments like tractor and all sorts of things came up, then we thought this will help us to dig the soil, because earlier the wooden plow, or what we technically call as moldboard plow, they were scratching only surface 3 to 6 centimeter of soil, not very deep, and then that used to result into some difficulties, of course. But when the tractors and the modern equipments came, then we thought that probably it will help us to plow the soil, make the lower soil upturned onto the top level, and it will help in increasing the productivity. But we found that it is not always true, because there is a increased erosion by breaking the soil clods and other things through the modern machinery. You are increasing the erosion, and especially in the windy areas like the Dust Bowl of central USA, or even in western India, for example, where huge wind can take away soil. There is a lot of surface soil loss because of the deep plowing. And also there is a water loss, because once you break the clods and more open the soil together, then the moisture retained and the lower layer, they are also lost. So the soil after some time becomes more dry, and ultimately bone dry rather, if you pursue with a very intensive system of modern agriculture. Now, there is also, it involves the principles of managing the topsoil. So the concept what we came, these are all learning processes. Actually, once we thought that this will be good, we saw it is not good, then we saw how we can avoid it. So this involves, conservation agriculture involves principle of managing the topsoil to create a permanent organic soil cover that will allow for growth of organisms within the soil structure. The third principle is practicing diverse crop rotation or crop interruption. This is another important thing. Most of the cases, say for example, the north and northeast part of the country, you have got cereals followed by cereals. That means you grow rice followed by that wheat, and then again rice. There's the same cropping pattern, and that leads to several harmful impact onto the environment and soil together. And so the practicing what is suggested is that diverse crop rotation. You grow cereals followed by maybe oil seeds crops, followed by vegetables, and introduce different crop interaction that makes the soil more healthy. Now, when you have got intensive farming practices, then there are three harmful impact on basic soil ecosystem. Is that the most visible is plant parameter. You grow intensively, so after some time the plant starts looking unhealthy. There will be infection of the pests and diseases and all sorts of things. Then that's one point. If you go on continuously farm and in the farming in the same pattern with the same this thing, it affects the plant parameter, soil nutrient status. Continuously growing the crop in the same soil will exhaust all the nutrients, available nutrients for the plant. So ultimately plants will fall sick. Then the third thing is that microbial nitrogen cycling guild. This is very important because these are natural things present in the soil which helps the uh, plant to grow in a very stable atmosphere. But once you apply some fertilizer and other chemicals, this natural N cycling guild is destroyed or disturbed, so that the nitrogen fixation and other things are affected. And then ultimately this leads to the loss of fertility of the soil. So when you have, I would say, would have told it as impact of intensive farming practices. These are some of the things which we always see when we do intensive farming year after year in the same cropping system, same pattern. Then these are some of the things which turns up. Now, a growing body of research is highlighting the impacts of root-associated microbial communities that can have impact on plant health and development. These impacts can include changes like yield in quantity and quality, timing of key developmental stages, and tolerance to biotic and abiotic stresses. So these are the things, impacts. This can be changed by the root associated microorganism. With such a range of effects, it is clear that understanding the factors that contribute to a plant beneficial root microbe may prove advantageous. Now, the functional importance of the plant microbiome. What are the functional importance? Is that one, mobilization and acquisition of nutrients. The microorganism, they mobilize the nutrients and also helps in the their acquisition for delivery or supply to the plant. You can see here a picture whereby you have got two plants. One is a little bit less grow, the leaves are not that wide, another broad. Another case, you have got a comparatively taller plant as compared to this, and the leaves are so big. And this is actually, one is inoculated with the plant growth promoting rhizobacteria, another is normal growing. We did not add any this thing, plant growth promoter rhizobacteria. So this functional importance is mobile. This is because of the mobilization and acquisition of nutrients. Also, there are so many other supporting roles. Is that out competition and antagonism of pathogen? They allow they outcompete the pathogens, these microorganisms which are beneficial, and they also act as antagonistic to the pathogens. So the pathogens cannot grow, they cannot thrive, or their population is kept at a very low level so that they cannot cause diseases to the plant. Then plant stress alleviation. Now this plant, the microorganism present in the environment, they can help the plant to avoid, alleviate the stresses. I will come to that with specific examples subsequently. Then there is a plant growth promotion. You can see between these two examples, there's a promotion of growth, co-metabolism, and degradation of toxins. So these are some of the things, the functional importance. Now, I am showing you the role of a plant and the bacteria through some graphics. You can see here this plant growth promoting rhizobacteria, or what we call as PGPR. This is a plant example. Actually, this is an Arabidopsis model, but it's just taken as a model of a plant. And you can see here, when you inoculate them, this plant with PGPR, PGPR produces some molecules which helps in the growth, better growth of this plant. Now, this is about the nutrition and growth. That means they provides the nutrients and the other things so that the plant growth is better. There is also the resistance or tolerance. Now is that the microorganism, this PGPR, they produces certain molecules which helps into what we call as the biobubble. Nowadays that biobubble has become very important, especially for people going abroad in this COVID time. Going abroad, they are, or the cricketers who came recently, like English cricketers, they are kept in a bio bubble. Means their outsiders are not allowed to enter, and they can be more remain in a more protected condition. So here, which are not allowed to enter is that the different pathogenic harmful microorganism. Here we have given a model of with a red, I mean dangerous color of this thing. So the plant is protected from this pathogenic microorganism. These are the resistance or tolerance. Also, they protect the plant from different environmental stresses, like say here I have put a bright sun to indicate that the plant is protected from drought, stress, and other environmental stresses, abiotic stresses. It can protect, and so the plant grows better. Now, when we put both the two cycles together, that is the nutrition and growth and the resistance of tolerance, then we find that there are some microorganisms which have got dual role. They also serve as nutrient or growth supplying as well as resistance and tolerance, and they are all characterized by specific metabolomic function. They have produces different types of chemicals, different types of metabolites, and that helps the plant to produce. Like here, these group of pathogens which have got resistance or tolerance, they produce salicylic acid, HMB, and different proteins or other metabolite by which they can protect the plants from the different pathogenic microorganisms. Now, nutrition and growth, they can produce some growth hormones like indo-lactic acid, then gibberellic acid, etc. And or they can help the plant to grow. These growth hormones, they will produce the plants to grow. But in between, you have got some of the compounds like bacterial volatile organic carbon like acetone, deoxys, or the node factor, which helps in the development of the nodule. Then pyvardin, these are the fluorescent pseudomonads, they produce as the pyvardins, and that helps in the protection of the plant from the diseases. And DAPG, fluoroglucinol, actually, they are produced by the pathogenic, they are produced by the organisms, and that helps in the growth, better growth of the plants. Now, whereas physicochemical properties of soil types, they determine the composition of soil biomes, plant root exudates can create an environment at the rhizosphere that gradually alters the soil biomes to favor the establishment of a rhizobiome. So this is actually a crosstalk takes place between the microorganism and the plant, that there is a direct interaction, direct talking. We call it crosstalk between the organism and the plant, is that they determine the composition of the soil biomes. Now, if you think of the soil physicochemical properties, each and every soil, the microbial properties are different. You take the laterite soil of the northern part of Andhra, or the black cotton soil in the western side. They have got basic soil. The soil biomes are different. Now, when you introduce the plants into those soils, then the plant provides an environment. They create an environment in the root region that gradually alters the soil biome to favor the establishment of a rhizobiome, that is microbiome in the root region. They release some of the organic substrates known as the root exudates, which are nothing but the photosynthesis produced by the plant. They supply it to the root to the microorganism, like we give food to the guests when they arrive. So here these guests are actually the different microorganisms, and this plant, they produce the root exudates, and that root exudates allow the proliferation of the specific group of microorganism being readily metabolizable carbon compounds. The bacteria microorganism use them, and they actually favor the establishment of a rhizobiome or a rhizosphere microflora, which is very specific to a plant. A rice plant will have a different type of rhizosphere microflora, while a Cajanus cajan or arar plant, a leguminous plant, will have a little bit different type of, or maybe completely different type of microflora. Now, these root exudates, together with the root immune system, would finally select for those microbes that have developed mechanisms to colonize the root rhizoplane, or sometimes they even enter the plant into the inner root tissue, which we then call as the endospheric microbes. Microbes we call. They remain within the plant, like somebody in the colony and somebody in your own house. You rent a upper floor, ground floor to a fellow. So he is inside the house. While in the society, in the colony, there are several other houses where people stay. So this is actually some sort of that type of allegory. You can use it, that they evolve mechanisms to colonize the root rhizosphere and the inner root tissue. Yeah, the interactions between plants, fungi, and bacteria in the rhizosphere, if you would like to check it, then you will see that there is a very close interaction between the plants, fungi, and bacteria in the rhizosphere. Now, one more thing is not added is that, of course, this is beyond the scope of this. I think I told in the last lecture, the soil animals. Soil animals are also present in the rhizosphere, and they have their own role in the cycling of the residues or detritus. Now, if you think of in this restricted scenario of plants, fungi, and bacteria and interaction in the rhizosphere, we can start with the plant. There is the rhizosphere, and in the rhizosphere, you see the black going away are the things which are produced and released by the plant. The root exudates, they contain phytochemicals and organic acid, and they supply it to the bacteria. This is a bacteria which grows with a flagella modular things. Root exudation contains phytochemicals and branching factors. So some of them are the signaling molecules, and these signaling molecules help in the growth of the fungi and the bacteria. And this actually also produces secondary metabolites like polyketides and other type of compounds which are produced by microorganisms. And those are, so the plant provides the energy, and the phytochemicals, etc., or the core carbon compounds, and the microorganisms, they produce the secondary metabolite. And these things, then what they do, this is a signaling molecules, they have a very tight interaction between the fungi and the bacteria. Now they actually signal molecules like auxin, cytokinin, ethylene, etc., supply to the plant. The plants grow because of the growth hormone present. Now this whatever is supplied to the bacteria, they have got the signaling molecules, they produce like peptides and other things, and they grow. There is a growing growth and the cycling there. Now they help in the quorum sensing molecule production, deleterious toxins, enzymes, etc., etc., to treat the, help the plant to grow. So these deleterious toxins means they actually help in the control of this production of the deleterious toxins. Sometimes they produce some antagonistic chemicals which helps in the control of the pathogenic microorganism. So this is the interaction between the plants, fungi, and bacteria in the rhizosphere. Now, if you look at the future, did I miss some slides? Okay, I'll come to that. Looking at the future, the functions of the plant microbiome are essential for the host. They are essential, this plant microbiome. Specific enrichment of microorganisms in plant-associated communities exists. Origin of plant-associated bacteria is different. Obviously, since the root exudate controls the plant, different types of plant species will have different types of root exudates, and that will allow the plant associated bacteria to proliferate. Horizontal gene transfer contributes to plasticity and evolution of plant associated bacteria. Now, plant microbiome discoveries could fuel advances in research on sustainable agriculture, such as development of microbial inoculants as biofertilizers, biocontrol, or stress protection products. In the future, the plant microbiome will have a greater importance for plant breeding and plant biotechnology. This is interesting because a plant and a bacteria have developed a association over several hundreds or maybe thousands of years that a plant is normally growing in the soil. Now, like a human being has got in their stomach, there is, we call it gut microbiome. You might be knowing it. Intestine, there is a microbiome. And if you remove all the bacteria from a human being, their weight will be reduced by almost maybe around a few kilograms. They will be reduced. The plants also, these microorganisms in the plant rhizosphere also a very effective relationship they have. Sometimes they are inside, as I said earlier, they are inside the plant, and when you remove them, those bacteria, what we call as the notabiotic culture, GNO, that means there is no other microorganism present in the plant, only the plant is there. That will look very sickly, that will not grow, and after some time they may die also. So that indicates that primarily these organisms, they have got a very close relationship. Now the plant breeders and the plant biotechnologists, they are looking at this issue and then trying to find out or rather breed the plants with the same microorganism, so that you have got a better type of plant available, which is already having a rhizospheric association. So it is suggested that the beneficial aspect of the entire microbiome should also be integrated as a biomarker. Now, plant microbiome can be an important source shaping other microbiomes. That's what obviously we are told, that by comparison of microbiome structure, a meaningful overlap of phytopathogenic diversity can be recognized among microbiomes, which are in the same way linked to each other. This may also include the human habitat and plants. So if you grow a plant inside your garden and a plant growing freely in the field, there could be differences in their physiology, physiognomy, as well as the microorganism present in the rhizosphere. I think I missed some slide. Let me go back. I missed some slide. How it is? I think there is some problem. Okay, TK, I think now I cannot go back. There is some problem in the slide. I think some slides got deleted. How, I do not know. So this is interesting in the sense that you have to have a very intricate relationship between the plant and the microbes. Actually, there are some slides on the, can I quit and then try to check that slides again? Stop share. Just a minute. Yeah. No, I think I got a different slide. Yeah, I think I went for a different this thing. Sorry, I will again join that one. Share screen. It was open. That's why it got. Just a minute. I'm sorry for this confusion. It was my creation. I should have checked it at that time. Okay.
सर कोई
So,
बात नहीं सर, ऐसा ऐसा मत
no,
बोलिए.
it is.
नहीं, इसमें भी तो इन्फॉर्मेशन बहुत सारा था, ऐसा कुछ
No,
नहीं.
no, no. I wanted to, what I wanted to show that it has gone out. I that time itself I checked this thing. Okay. Now I think I showed here no, up to this. So let me just. Is it visible now?
हाँ हाँ दिखाई दे रहा है. Yes, it is visible.
Okay, okay. So this has to go share. It is okay. Then how to remove this? How to go to full screen? It is not visible here. Yeah. Right. So previous. I think I missed from here only. I went to the other slide. That's why. Okay. Now the plant growth promotory microorganism. I think I should have it should have come to earlier. There could be different things. There could be the mobilization of the different plant. I mean indirect mechanism, and there could be direct mechanism. Direct mechanism is that they produce the biological nitrogen fixation, nutrient mobilization and acquisition. I have told phosphate and potassium and even zinc, etc. They will mobilize to the plant, and the plant growth hormone production, different types of plant growth hormone production. And there could be lowering of the pH through organic acid and chelation of cations. This also should be there. Then their indirect mechanism could be through antibiotic production, starch hydrolysis, hydrocyanic acid production, cellulose degradation, H2S production, siderophore, and antifungal uh, activities. Now, among the diazotrophs, you could have both symbiotic and associative asymbiotic organism. A symbiotic organism, the most common thing we know is already the rhizobium or the rhizobial group. There could be different groups of the organisms they are now classified onto alpha proteobacteria, beta proteobacteria, and actinobacteria. And examples are the parasponia, the leguminous bacteria present in the legume root nodules, and the actinorhizial plants like the Frankia, you get it in Casuarina, etc. Also, there are cyanobacteria which are present in the cycads or the even your azolla, you get cyanobacteria, etc., etc. Basal eudicots also sometimes you get cyanobacteria in the root region. Now, there is also associative asymbionts like the gamma proteobacteria, then present in these are the gamma proteobacteria. They could be present in the rhizosphere or could be in the endosphere, and they're present in cereals, ferns, bryophytes, etc. This I have already told. Sorry, I got this thing. Now, in addition to these properties, what I have shown earlier slide, the previous slide, in addition to their properties on the growth and the plant protection from the diseases, sometimes they also help in different way, like selective fungi and bacteria, which symbiotically colonize the roots of diverse plants. They can increase the plant's photosynthetic capabilities by reprogramming the gene expression. Now, this is very interesting, like because photosynthesis is the major process of the plants that actually they fix the carbohydrate or the carbon. Sorry. They produce this carbon, and fix the carbon, and this photosynthesis actually is conferred upon the some of the fungi and bacteria. They actually, this endophytic in endophytic oh, these endophytic bacteria, they actually help in the means stimulation of the photosynthesis of the plants. Now, this is conferred by commonly exhibiting numerous advantages over similar plants not colonized. When the plants are not colonized by those specific organisms, then they have got lower photosynthetic potential in comparison to those plants which are inoculated or contaminated by those organisms that can help. Now, how they do, there are different mechanisms suggested. One thing is that the photosynthetic bacteria or the microorganism, they help with their apparatus for the fixation of carbon photosynthesis system, because the bacterial photosynthesis is different from that of the plant photosynthesis. So that could be one way. Second thing is that sometimes the under stress condition, these photosystems, they result into the production of the reactive oxygen species. You probably know, colleague, I mean, attendees may probably know that the RUBP carboxylase, which is helps in the fixation of carbon dioxide under stress condition. I mean, this RUBP carboxylase is an enzyme which goes on working in the presence of light and energy, and then they go on, they don't bother for whether you have got the carbon dioxide or not. They go on producing the oxygen, reducing power. Now, when the oxygen is, when the reducing power is restricted, especially under drought and all, drought condition and all, then what happens is that they converts into oxidative processes, and they produces what is that reactive oxygen species, which is damaging to the plant organs, because they can oxidize the proteins, lipids, and all the different enzymes. So these reactive oxygen species, they actually are produced under specific abiotic stress condition. Now, these microorganisms, they help in the tackling of these ROS, like certain trichoderma species. They activate the biochemical pathway that reduce ROS to less harmful molecules like catalase, peroxidase production, etc., which help in the catalysis of the ROS, reactive oxygen species, and that is converted into H2O, and it becomes harmless. So under that condition, the photosynthetic machinery is protected and allowed to function better. So this indicates that these microorganisms can also help some another way as compared to the commonly known processes of plant growth support, like producing hormones or producing mobilizing nutrients or the antagonistic moieties, etc., etc. Now, this I think I have already told. Of course, these are now, it doesn't have any importance since whatever I have spoken is that, like the microorganisms, they act as a biofertilizer. I have mentioned them. Some of them, they produce the different compounds, and the bacterial biofertilizers, there are several ones like Azotobacter, Frankia, Rhizobium, Allorhizobium, etc., etc. They help by producing phytohormones, siderophore, nutrient enrichment, improved photosynthetic performance. That's what I have told just now. Improved plant tolerance to stress and resistant to pathogens, increased productivity, and crop improvement. Now, they also, the nitrogen cyclers, they actually helps in the production, fixation of nitrogen to ammonium, ammonia to ammonium, and that is taken up by supplied to the plant. It could be that way, or they can be converted into nitrite, nitrate, and even maybe denitrified to N2. So a proper balance is maintained. Also, there are fungi like mycorrhizal fungi, which remains inside the plant root, and they help in the solubilization of phosphorus and supplying it to the plant tissue. Also, because of the endophytic growth, they increase in the volume of the plant tissue, and help in the water relations of the plant. They can retain some plant during the drought season, etc. So they can help in several other ways. So this, what I want to mean is that by all this diverse example is that it is not only the plant growth promotion, but also a diverse role the microorganisms have for benefiting the plant. Some of the commercial products are developed using the different PGPR strains. These are all, unfortunately, in my slide, these are all international brands. This does not mean that we don't have. In India also, several companies have come up now which produces the commercially, they produce the different PGPR strain and supply it to the farmers. So like Agrobacterium, Radiobacter, there are some names. I think these names are not of that importance, but as I said, these are mostly international brands. Some of my friends gave me this slide. They can be Azospirillum brasiliensis, Bacillus subtilis, Bacillus amyloliquefaciens, then Burkholderia cepacia, Pseudomonas fluorescens, Pseudomonas syringae, and Streptomyces griseoviridis. So these are the different PGPR strains which help in the have been commercially developed using PGPR strain. Now there are several big companies now which multinationals which produces this PGPR strain and supplies to the farmers. This I missed in the last slide. Others could have been told anyway. Microorganisms providing tolerance to stress, and as you know, there could be two types of stress: abiotic stress and biotic stress. Abiotic stress is that because tolerance to drought, tolerance to submergence. Drought means obviously when there is less moisture or moisture deficit. Tolerance to submergence when moisture is in excess. Tolerance to salinity. So this the biotic stress also you have got systemic acquired resistance and induced systemic resistance. These are the two types of resistance a plant expresses when they are exposed to biotic stress, and these are different different metabolic pathways, biochemical pathways by which one is ethylene mediated, another is jasmonic acid mediated. So I'm not going to all those details. Like here I am showing you the rice seedlings exposed to submergence stress under autobiotic condition. I mean, when we grow the normally a rice seed, even if you put you underwater, it will germinate, but it will not grow because there is no oxygen. And when you have got a standing water above the this thing, the solubilization of the water oxygen is very low, almost 1 by 1000 in air. So in aerobic soil or ordinary dry soil, the air that goes inside the soil, they contain a lot of oxygen. But when you have got water, like in a rice field, then the oxygen cannot grow inside. The soil is mostly anaerobic. I'm in a meeting. Please follow this. So this, like here, when we have inoculated the rice plant with certain bacteria, then we find that they can grow even maintain under the submergence. Otherwise, normal condition, they will die. They will germinate, but after 6-7 days, they will die, like here. And then here, they will not grow. But when you inoculate with some bacteria, then specific bacteria, then they can grow. And we have found the exact reason why it grows, that they have produces an enzyme ACC deaminase, and ACC deaminase, it reduces the destroys the ethylene. There is no ethylene production, which helps in the plants to grow, because ethylene is a growth inhibitory hormone for mostly decay and other purposes. So this ACC deaminase containing rhizobacteria, they help the plants to grow even under submergence. Now this is submergence, and when there is salinity, I have got another. You can see here, this is again some rice seedlings, and they are exposed to salt stress. This salt stress actually 150 millimolar sodium chloride solution we have given, You can see this is a, when you give the salt stress, then they do not grow. Only they remain like that. But when you inoculate them with a certain bacteria, here, this is actually, C is a heat killed same bacteria, whatever we have grown here. This allows the plants to grow. Here the plants are growing. So even in a saline condition, you can see here also, there are actually 3 microorganisms are there, 3 or 4 bacteria are there we are testing, which is most useful. They can help the plant to grow even under salinity stress. So this is the abiotic stress in which they can grow. So I think this I have already told. Now, one I would like to go back again these prospects and uh, uh, now you see the microbes can fulfill diverse bacterial interaction in plants, leading to promising solutions for sustainable and environment friendly agriculture. What are those? Improved understanding on the way by which microbes promote plant growth that can lead to expanded exploitation of these biofertilizers to reduce the potential negative environmental effects associated with chemical fertilizer. We have I think I missed that slide because of the confusion. You know that when you continuously apply chemical fertilizer, first of all, it reduces the potential of the chemical fertilizer when you go on applying repeatedly. Our, I mean, old farmers, they will tell that when they started initially applying fertilizer, they used to get so much of yield. Now applying so much of fertilizer, they don't get that much yield. We have made some calculation, like 1970s, when the chemical fertilizer was first introduced. Our rough calculation was that 1 kilo of nitrogen fertilizer will give to 20 kilogram of grain. That was the rough calculation in the late 70s when the chemical fertilizer was introduced in the country on a large scale. But nowadays, 1 kilogram of chemical nitrogen fertilizer will give you around 6 kilogram of grain. So you see, over the years, use of chemical fertilizer, the efficiency has been reduced. You are not getting the same effect. Also, continuous chemical fertilizer application will result into the loss of organic matter of the soil. The soil will become dry, and also salinity will also come. So these are the environmental effects. I think I missed that slide beforehand. So improved understanding on the way. The second thing is that sustainable agriculture, such as development of microbial inoculants as biofertilizers, biocontrol, or stress protection products. This I have already given the example. The rhizobacterial community can be specifically engineered to target various functions to improve plant productivity. Now, recent progress of molecular biology and biotechnology in the understanding of rhizobacterial interaction with the nodules of crop plants will encourage a suitable area of research in PGPR mechanism relating to rhizosphere colonization. Many laboratories all over the world are now trying to explore what is the exact mechanism. We say that a bacteria is inoculated, you inoculate, there is a good impact. Now, why? What are the exact things? We find that apart from the so-called beneficial effect like growth hormone production or the nutrient mobilization, etc., they serve the plant in so many other interactive ways, and that is what is the crosstalk between the bacteria and the plant, which are now being explored. Okay, now I am giving you example here, some sort of a sustainability, including both the microbial fertilizer as well as the chemical fertilizer. You have got 3 plants here, some model, of course, this is not any typical plant. You see, chemical fertilizer, when you apply chemical fertilizer, these are the white grains are the chemical fertilizer grains. This chemical fertilizer, and when you apply, a major part of the chemical fertilizer is lost to the air, volatilization. And of course, they increase plant growth, they increase yield, but at the same time, there is a poor soil organic matter and fertility, high nitrogen loss, less root exudate, low microbial activity, and provide only specific nutrient. Although it is written only N, it could be specific nutrient to the plants. When you apply the microorganisms only, these are the, now here you see the number of microorganisms are much less when you apply the chemical fertilizer. Now when you apply the, don't apply the chemical fertilizer, there is a large number of microbial population, highly diverse population, and because there is no concentrated chemical fertilizer, so the few N losses to the air, and they fix atmospheric nitrogen, they provide other nutrients, they increase the soil fertility, they do not harm the soil. So this is the thing. Although the yield may be at par or slightly less, they have got several beneficial effects. Now, in order, if we can combine these two, you give half dose of chemical fertilizer plus diazotrop. Here, chemical fertilizer is also there, half dose, but these are the conventional chemical fertilizer. These are the modified chemical fertilizers. And then what will happen is that this chemical fertilizer will not be lost immediately. They will be slowly released and supply the compound to the plant. So they will not at a time affect the harmful effect on the soil microbial population. So you will get increased growth and yield, then root proliferation, higher uptake of water and nutrients, better SOM, few or no N losses to the soil, no pollution in the soil, higher root exudates, higher microbial activity, provide all the nutrients and nutrients to the plant. So this is a strategy that is being developed, that you harness the maximum from the plant. You can see here between these two stature of these two plants, one case there is a huge growth, better growth as compared to the only chemical fertilizer. So this is one thing, and then this also helps in the a more sustainable environment, whereby you have got less this thing. Now I wanted to tell you the approaches to genetic and genomic engineering of phytomicrobiome. One is, we call it a bottom-up approach, where we isolate the microorganism, identify them, and then finally identify the desired trait to put into the, through transgenic mechanism, we put into the plant, and they get the desired trait inserted into the plant. This is the bottom-up approach. In top-down approach, it is uses the gene transfer to introduce traits into broad range of hosts, and then screen the host for a better phenotype by using the other supporting devices and omics technologies, etc. So this actually, these are the two mechanisms that one can follow, either bottom up or the top down approach. Now, the main benefits of sustainable agriculture, if we tell, and the principles of sustainable agricultural practices, is the enhancement of soil quality. To come back to the, in the beginning, what I started with, enhancement of soil quality, fertility, production, and amelioration of soil physicochemical characteristic, improvement of plant physiological status, increase of the abundance, functionality, and diversity, re-establishment of the trophic balance, and preservation of natural resources. And the examples of sustainable agricultural practices, we have already I have already told actually in the beginning itself, that less tillage, this I have introduced, cover crop, then cycling of the different crops, etc., etc. So I would like to close my lecture by showing this slide, is that future of the earth lies in scientific management of its resources. I mean, we should not be greedy, that we will use all the resources. This earth we will live, but we will go, but the next generation and next to next generation will come, and we have to keep them habitable. So only hope is that we manage the resources in a scientific manner, and thereby we continue to maintain this earth more sustainable, more green. Thank you.
Dear participants, if you have any queries, please put questions in chat box. Prasanna, okay. Prasanna, you can
Good
ask
morning.
question, Prasanna.
Yes, sir. Good morning, sir.
Good morning.
Sir, you were saying about trichoderma in salt stress, sir.
Yeah.
Trichoderma in salt stress. So will it work out for the temperature stress also, sir? If we give highest temperature like 45 degrees, will it will this temperature harm the microbe? Or can we work for a rhizomatous plants, sir? I'm asking.
See, I think I could not very clearly told you. I was taking trichoderma as an example, but then trichoderma has got beautiful potential. They have got several functions. Yes, they can help the plant to grow under stress. Especially, I mean, several field experiments have been done whereby trichoderma has been shown. It has been shown to provide the plant with properties to alleviate the stresses like drought, or sometimes to certain extent high temperature tolerance. It is there. So one has to really test it and then provide the information. It can do wonders. Trichoderma is considered as a wonder bacteria. There are 4 species, of which Aspergillus is a very common one and has been used extensively for development of formulations, and especially for protection of the crop from the pathogenic microorganism also. So it could be possible. I mean, I have not worked with trichoderma, so I cannot specifically give you the example, but then it is there, it is known.
Thank you, sir.
Further, anyone want to have a question, please? You can raise the hand, or you can post in chat box. As of now, as there are no questions, thank you so much, sir, on behalf of the Department of Botany and whole participants of this 2 weeks refresher course. Wholeheartedly, we express our deep gratitude for such wonderful presentations on both the topics and motivating our teacher friends and the scholars.
Participants are requested to follow the instructions during session.
Shivanna sir, can you start sharing the screen?
Video to avoid disturbances.
Good morning, sir. Shivanna sir.
Post your queries in the chat box only.
Yes, I can start.
Do not unmute your
You start sharing screen, sir.
session unless
Yeah.
you are asked.
Shall I share the screen now? Hello?
Yes, sir, you share the screen.
So now which one we have? I share the screen. Now which one I should press now?
Sir, down you have green arrow up, share screen.
So share screen I pressed. Then
Now you will see your desktop. If you have opened your PowerPoint, you have to go and click that PowerPoint.
okay, I clicked the PowerPoint. Then shall I share? Okay.
Yeah, it is coming. Yeah, it has come.
It has come. Now I will make it full screen. Now anything else I have to do? So can I put it partial screen on one side? Is it okay now? Hello?
Fine, sir. It is fine.
So once again, I would like to greet everyone here.
So I want to remove this side one. Can I remove this side one where the full screen can be seen, or can you put that screen on one side only? Dr. Khan. Is it okay? Can I start
Sir,
now?
yeah, yes, sir. Yes, you can start, sir. Participants, we have again back with our Professor K.R. Shivanna, sir. He will be teaching us today about how do flowering plants select suitable pollen grains for fertilization. It will be again a wonderful session for us with her. Sir, you can begin your talk, sir.
Okay, friends, I am glad to participate in this webinar of the refresher course for teachers. And today I will be talking about how flowering plants select suitable pollen grains for fertilization. As many of you would be knowing, pollination is the first step in which male partners and female partners are brought together. And out of that one, how the right type of pollen grain is selected for fertilization. Yeah, why it is okay. Now, for fertilization of male partner, pollination is the prerequisite. So the selection of the male partner in flowering plants is in fact more complicated when compared to any other groups of plants. Pollen grains, as you know, are the male partners. They are produced in the anthers, and the ovules, which are present in the ovary, which is a part of the pistil differentiated into the stigma, elongated style, and ovary, they are the female partners. Now, selection of the male partners starts with pollination. So pollination means transfer of pollen grains from the anther to the stigma. In very rare cases, the pollen grains are deposited on the stigma of the same flower. So self-pollination is rather rare. It is mostly cross-pollination. That means the pollen grains come from other flowers, either of the same plant or of different plants. So pollination is the first and the most critical step to bring male and female partners together. So until pollination takes place, then the male and female partners do not come together at all. Now there are different types of pollination. And these are, for example, if the pollen grains of a flower get deposited on the stigma of the same flower, it is called autogamy. Whereas if the pollen grains belonging to another flower located on the same plant are transferred to the stigma of another flower of the same plant, it is called geitonogamy. When the pollen grains coming from the flower of situated on another plant, if they fall on the stigma of this plant, that is called xenogamy or true cross-pollination. If two plants happen to be the clones, even if the pollen grains coming from another plant technically, but because they are clones, even if they fall on the stigma of this plant, it is continued to be called geitonogamy because it is strictly a self-pollination. Now, plants are sedentary. They can't move from place to place. So there is no way for them to carry the pollen grains and put them on the stigma. So what they have done is they have outsourced this pollination function to other agencies. Now, which are these agencies? About 90% of the flowering plants, they use a range of animals to bring about pollination. Whereas the remaining plants use either wind, most of them, or very few of them will use water to bring about pollination. Wherever animals are involved, of course, it is called biotic pollination. And where the wind and water is involved, they are called abiotic pollination. So one thing is, as far as pollination is concerned, the pollinating agents, whether they are animals or whether there is wind and so on, there is no way that they will bring only the pollen of the same species. They may bring pollen grains either from the same flower or from another flower of the same plant or from another flower situated on another plant. So the pollen grain that the stigma receive more often is a mixture. So sometimes what happens is, whether it is wind or whether it is any of the animals, if they happen to visit the flowers of other species and visit the flower of this species, they may bring the pollen grains of other species also. That means both homospecific pollen and sometimes the heterospecific pollen may get deposited on the stigma. So here, amongst the homospecific pollen, as I told you, there may be two types. One is coming from the same plant, which is called autogamy, or it may be through veterinogamy, and the pollen grains may come from another plant, which is called xenogamy. In heterospecific pollen, the pollen grains may come from a species which is very closely related to this species, or the pollen may come from species which is far related from or distantly related from this species. So as I told you, more often pollen received on the stigma is a mixture of different species. And this is particularly true in wind pollinated species. So as you know, any organism, whether it is a plant or animal, whatever the case may be, if it has to maintain the integrity of the species, fertilization has to take place between gametes of the same species. Because if the gametes of different species start fusing, and if they develop into plants, then it becomes nonsense, because the integrity of the species cannot be maintained and the evolution cannot proceed normally and so on. This is one of the basic aspects of fertilization. It has to come, the fertilization has to take place between gametes belonging to the same species. Now, let us see how this selection between gametes of the same species is ensured in different groups of plants. Now, I have just tried to put it in a simple diagrammatic way, how the gamete recognition to make sure that the gametes coming from the same species are fused is ensured. Going to algae and fungi, for example, let's say this is a sporophyte. You please, for today's lecture, you forget about S1, S2, and so on I have given, because I don't have the time to explain all those things. So this is a sporophyte. And the sporophyte, whether it is isogamous or anisogamous, it releases the gametes into the aqueous medium, surrounding aqueous medium. So these gametes will come in contact with each other. They may be produced by the same sporophyte or different sporophyte, or those gametes may come from other species which have released these gametes into the same aqueous body. Then here, the recognition, when once the two gametes come together, they establish recognition. If they come to this, belong to the same species, they recognize each other and fuse. If they happen to come from different species, recognition cannot be established, and then they don't fuse. So that is how here the gamete recognition is established at the level of the gametes, whether it is isogamy or whether it is anisogamy. When we come to bryophytes and pteridophytes, here, of course, male gametes are released into the surrounding aqueous medium, but female gametes are retained in the archegonium. Now here also, although female gametes are retained in the archegonium, the male gametes are able to swim through this, the neck, and then reach the female gamete. Here also, recognition takes place at the gamete level, and during this fertilization, it is ensured that only gametes coming from the same species are involved in fertilization. Whereas when we come to gymnosperm, here the whole male gametophyte along with the gametes is enclosed in the pollen grains, and these pollen grains are discharged into the atmosphere. And the female gamete, along with the female gametophyte, is retained in the ovule, that is a sporophytic tissue. And here pollen grains, which contain the vegetative cell, and finally the gametes, they come in contact with this tip of the ovule, the pollen chamber. There they will germinate, and the resulting pollen tubes have to pass through a few layers of the sporophytic tissue before they get into the archegonia or the female gametophyte and discharge the male gametes. There the male gametes will come in contact with the egg and fuse. Now, until now, what we don't know is when the pollen tubes pass through a few layers of the sporophytic tissue in the ovule, whether there is any interaction or not. So whether in gymnosperms, the gametic recognition takes place at the level of this sporophytic tissue or at the gametic level itself, as it takes place in the earlier lower groups of plants. So by seed plants, whether it is gymnosperms or flowering plants, they have become independent of water for fertilization by evolution of the pollen grain. Because here, because of the pollen development, they don't depend on water for fertilization. Now, when we come to flowering plants, it is much more complicated fertilization. Pollen grains, instead of landing at the tip of the ovule, as in gymnosperms, they land far away from the ovule, that is on the stigma. Now, on the stigma, they will germinate, and the pollen tubes have to pass through a long distance through the sporophytic tissues of the pistil before they reach the ovule. So there is a massive gametophytic and sporophytic interaction before the pollen tubes can enter the ovule and discharge the male gametes. Now, all the evidences show that the recognition of the pollen or screening of the pollen or selection of the right type of pollen takes place during this sporophytic and gametophytic interaction in the pistil. So it appears that the function of gamete recognition probably has been transferred from the gamete itself to the sporophytic tissue of the pistil as far as flowering plants are concerned. But as I said in gymnosperm, we still don't know whether these diploid neutral cells have any role or not. Now, first of all, as I told you, there are basically three types of pollen grains on the stigma. One is heterospecific pollen. That means some pollen grains amongst that group of pollen grains that are present on the stigma, they may come from other species. Now, how do these species, they have to be rejected. They should not be allowed to go through and affect fertilization. Now, to understand this one, we can visualize the whole pistil of flowering plants as the home of the female partner, that is the ovule, with a long corridor. And this corridor has locked doors at different levels of the corridor. One at the level of the stigma, there is another locked door somewhere at the upper part of the style. There may be another one in the middle part of the style. There may be another locked door at the lower part of the style. Now the pollen grains, if they have to go through, whether at the level of germination or at the level of pollen tube growth in the pistil, they have to have a matching keys to open the locks present in the pistil. Now, pollen grains, if they happen to be from the same species, they have all the keys to open these locks. But if they are coming from other species, they don't have the keys to open the lock. Because as speciation proceeds, the new species becomes gradually more and more distantly related. So depending on how far the evolution has taken place, how distantly the two species are related, then this the inhibition of this one may take place at the level of the stigma itself, or it may take place at the level of style, upper part or middle part or lower part, and so on. So basically, this is how the heterospecific pollen grains are rejected. There is no positive recognition simply because the pollen grains coming from other species don't have the matching keys to open the style at different levels, the locks at different levels, so they just cannot get into the ovule and achieve fertilization. So here I have just given some photographs of our work. Here you can see on the stigma, this is one of the crucifers. Although some of the pollen grains have managed to germinate, they can't get into the stigma. Look at this, this is stigmatic papillae, this is the pollen tube that has come out, but it can't get into the stigma. Now another combination, depending on the relationship between the two species, here the pollen tubes are able to enter the stigmatic papillae, but they can't grow further, they get inhibited. Here, they have managed to grow a bit into the style, but then they are inhibited. And here, of course, they have gone up to a distance in the style before they get inhibited. So this is how the rejection of heterospecific pollen takes place in general. Now, coming to the homospecific pollen, there are two types of homospecific pollen. One is self-pollen coming from the same species, same plant. And the other is cross-pollen, which is coming from another plant of the same species. Both these have the matching keys to the locks present in the pistil. Now, one thing we should remember is just because the pollen grain has a matching key, it doesn't mean that it will be successful in germinating and the pollen tube growing through the pistil and entering the ovule and achieving fertilization. In addition to the presence of matching keys, it has to be facilitated by the pistil. The pollen grains have to be recognized by the pistil and they have to be facilitated. The pistil has to facilitate their germination and tube growth. So if any recognition has to take place in the plants, then it has to be through chemical components present in the pollen grain and present in the pistil. Now, once we say recognition factors are in the form of chemical moiety, then the three things will become important. One is, where are they located in the pollen grains and in the pistil? What is the nature of these recognition factors? And what is the basis of recognition? These are the important aspects about if you have to understand about how the pollen grains are screened. Of course, now until 1970s, there is absolutely no information on these at all. But now there is some information, at least on limited number of species and so on. So I can't go into the very detailed aspect because the data is so massive, but I will try to give you a sort of a general idea as to how a pistil is able to screen the pollen grains. Now, coming to the location of these factors, which are involved in pollen-pistil interaction, which are involved not only in pollen recognition, but also in facilitating pollen germination and pollen tube growth and so on. As I told you, until 1970s, there was no information at all. But in 1970s, earlier part of 1970s, the information from pollen side came from Professor Heslop Harrison's laboratory, who was the director of Royal Botanical Gardens, Kew. And there, for the first time, some of his associates, particularly Bruce Knox, they showed that pollen grain wall contains extracellular proteins, both exine and intine. These extracellular proteins, they are dual in origin. The intine proteins come from the pollen cytoplasm itself and are incorporated during pollen development. And the exine proteins, They come from the surrounding tapetum, which is a diploid tissue. Now, these proteins are present in the pollen grains of all the species. Of course, it's a heterogeneous component. It contains proteins, glycoproteins, carbohydrates, some enzymes, some polyphenols, and so on. It is a complex mixture. Now, coming to the female side, we did not have any information at all until late 1970s. Now, in 1975, I went to Royal Botanic Gardens, Kew, for 1 year worked in Professor Tesla Patterson's laboratory. And during that period, I screened the stigma of 1,000 species and classified them according to stigma morphology. Basically, of course, there are details. I'm not going into all the details. Basically, there are two types of stigma. One is of the dry type, where you don't find any stigmatic secretion at the time of pollination. Another is the wet type, where you find some stigmatic secretion, whether very scanty or very massive as it is here at the time of pollination. Now, I found when I screened these, I found that all the species I have screened, that means about 1,000 species I have screened and classified them according to the structural details, invariably they contain extracellular protein. I could not only find out and I could generalize the statement that this is one of the unique features. All the plant species, they contain extracellular proteins on the surface of the stigma, whether it is dry type or wet type. In dry type, these extracellular components are present in the form of a very thin layer, which we called it as pellicle, outside the cuticle on the stigmatic surface, whereas in the wet type, they are present as a part of the stigmatic exudate. So whether it is the pollen grain or the stigma, there are extracellular components which are present on the surface. Now, so far, it has not been shown whether it is pollen or stigma of any species, which are free from these extracellular components. So this one, the data I have collected for 1 year in Royal Botanic Gardens, screening 1,000 species, published in Annals of Botany in 1997. Now it has been one of the highest cited paper ever published in that journal. So it has about 900 citations. Anybody who is working on pollen stigma interaction or stigma, they have to cite this paper. It has become a classical paper. Now the pollen grains, when they land on the stigmatic papillae, or whether it is a solid or hollow or a wet stigma, then they get hydrated and they release these wall components onto the stigmatic surface. Now these wall components from the pollen grains released onto the stigmatic surface, they will act as visiting cards. They are able to tell the stigma from which species they have come, whether they have come from the same species or they have come from different species. And this the extracellular components which are present on the stigma surface, they will be able to read these visiting cards and then recognize whether it is pollen is of the right type coming from the same species or it is of the wrong type. So if it is of the right type, then it has to activate physiological processes to facilitate pollen germination, pollen tube growth, and so on. Whereas if it is of the wrong type, they simply keep quiet. And if the wrong type, of course, in any case, it cannot get in because they don't have the suitable keys to open the locks present on the at different levels of the pistil. So now, coming to the self-pollen grain, that is coming from the pollen coming from the same species, there are additional complications of screening of the pollen grains. As you know, unlike animals, majority of the plants produce bisexual flowers. That means they produce both male and female partners. And also there is ample opportunity for self-pollination, whether it can be from the same plant flower or it can be from the pollen may come from another flower situated on the same plant. Now, if this goes on regularly, that will result in inbreeding. And this inbreeding continuously will result in inbreeding depression, which is not good for sustenance of the plant as well as its evolutionary potential and so on. So plants have evolved different mechanisms to prevent selfing. What are these mechanisms? Of course, I am telling you very briefly. One is dichogamy. Dichogamy means the temporal separation of pollen release and stigma receptivity. So in protandry, for example, pollen grains are discharged first, and the stigma will become receptive later when the pollen grains are no more available on this flower. So at least self-pollination is prevented. It doesn't prevent the pollen grains coming from other flowers located on the same plant. And whereas hercogamy means spatial separation of the anthers and the stigma. So in some hercogamous one, the level of the anthers may be much lower when compared to the stigma, or it may be opposite also. So self-pollination is to some extent prevented, but it is not prevented completely because pollen grains from other flowers may still come and land on the stigma. The other common mechanism, which is prevalent in a large number of species, particularly wild species, is self-incompatibility. So self-incompatibility means the self-pollination do not result in fertilization because the pollen grains are inhibited during germination, or the pollen tubes are inhibited during their growth in the pistil. This is self-incompatibility, and large number of species are self-incompatible, and this prevents inbreeding completely. Another method of preventing inbreeding is diclining. That means here the flowers are unisexual, but out of this one, they may be monoecious, where male and female flowers are born on the same plant, or they may be dioecious, where male and female flowers are born on different plants. So here we can say this is a male plant, this is a female plant. So technically, the dioecious plants or self-incompatible plants are strictly cross-pollinated. That is, inbreeding is completely prevented. Whereas in others, there can be some inbreeding, although it is highly reduced. Now, inhibition of self-pollen grain, that means when once the self-pollen grains, that is self-pollination takes place in self-incompatible species, the pistil will recognize that it is a self-pollen, and it will activate physiological processes to inhibit pollen germination and pollen tube growth. So to tell in a simplistic way, although these pollen grains have all the matching keys to open the locks present at different levels, but when once the self-pollen grains are recognized in self-incompatible plants, the pistil will see that these doors are bolted from inside by activating the required physiological processes. So the self-pollen grains, even if they have a key, they can't get in because they are bolted from behind. Now, of course, we have a lot of information about self-incompatibility. We know the genes which are involved in self-incompatibility, the gene products from male side as well as female side, how the recognition is established and so on. So that will be a separate lecture because there is so much of information is available. So I'm not going to that detail. So this is how self-pollen grains in self-incompatible plants are recognized and inhibited. Now, the acceptance of homospecific cross-pollen grains, which have the matching keys that is present in the pistil, they are technically all cross-pollen grains are eligible for fertilization because they have the matching keys. The self-incompatibility inhibition is not activated because they don't have, they are not self-pollen. So the cross-pollen grains are recognized by the pistil, and then the pistil will activate all the physiological processes that are required for pollen germination and pollen tube growth. So as I told you, technically all homospecific pollen grains are eligible for fertilization. Now, in general, what happens is the number of pollen grains that get deposited on the stigma due to natural pollination is many times more than the number of ovules available for fertilization. For example, you take cereals. There, each flower has only one ovule, but the number of pollen grains that deposited on the stigma may be hundreds. So sometimes in maize and so on, there may be more than 100. So out of so many pollen grains that get deposited on the stigma, only one of them will be able to achieve fertilization. Now the question is, which one of them will be able to achieve fertilization? Of course, here this is just a photograph to show how self-cross pollen grains, they germinate. Large number of them have germinated. This is a photograph of Nicotiana. The pollen tubes are entering the stigma. This is part of the style where the pollen tubes are growing very happily without any problem. And here, the pollen tubes enter the ovule. Then, of course, they discharge the pollen grains, the male gametes, and they result in fertilization and development of the seed and so on. Now, as I told you, the number of pollen grains that get deposited is many times more than the ovules available for fertilization. Then, as I told you, which of those will be able to achieve fertilization? Now, here is the beauty. When compared to other groups of plants, the pistil can organize as a Swayamvara. As you are all very familiar with the Swayamvara from our mythology. So it organizes a sort of a Swayamvara in the pistil. So what it does is it creates intense competition even amongst cross pollen grains. And out of so many pollen grains which are present on the pollen, on the stigma, only those pollen grains which germinate early, quickly, and whose pollen tubes grow faster, that means which are more vigorous, will be able to reach the ovules early and affect fertilization. So that means out of hundreds of pollen tubes available on the stigma, through this Swayamvara, the pistil is able to get the most vigorous pollen tubes that will achieve fertilization. Whereas those pollen grains which are less vigorous, that means it takes longer time to germinate and the growth of the pollen tubes is slower, will not be able to compete with vigorous pollen and will not be able to achieve fertilization. So they get eliminated. So this is the essence of Swayamvara that takes place in the pistil. Now, does it have any role in the fitness of these progeny? Of course, there are many evidences. I will just give you one evidences. There are evidences to indicate that such pollen competition in flowering plant increases the fitness of the progeny. So as I told you, there are large number of pollen grains. They may be able to germinate, but out of those, only a small proportion of them will be able to achieve fertilization. Now, in one of the systems, that's in Petunia, for example, people have been able to manipulate this competition. Now, as you know, in Petunia, there are about 100, more than 100 ovules, depending on the species in the ovary. So what you can do is one can reduce or eliminate competition by putting the pollen grains that are lesser than the number of ovules available. Let's say if there are 100 ovules, you put only 100 pollen grains so that whether they germinate quickly or later, or whether the pollen tubes are very fast or slow, they will be able to find a partner when they reach the ovary. Now, in another experimental pistil, you put much larger number of pollen grains on the stigma and create intense competition. So if there are 100 ovules here, you put 1,000 pollen grains on the stigma, and then you collect the seeds from both of them. And experimental studies have shown that this one, where there is severe competition, the seeds collected from this, they produce more vigorous and uniform plants when compared to the seeds which resulted with no competition at all. So this is one of the important thing we have to realize. Now, as you know, flowering plants evolved. They were the last to evolve amongst the plant kingdom. They evolved according to earlier concept. It was about 100 million years back, but now people are going back. Maybe some of the evidences indicate they would have originated about 400 million years back and so on. But whatever is the case, but they are the last to evolve in the evolutionary scale. But they became more successful, and within a short time, they became the dominant plant kingdom on Earth when compared to all other groups of plants. People have been trying what would be the reason about the evolutionary success of flowering plants. Then many people then argued, and there is general acceptance that evolution of the flower is one of the significant features. This has more biotic pollination, more effective. And out of this, amongst the flower, the evolution of the pistil, which is another factor in which the recognition and selection of pollen grains by creating intense competition, even amongst the compatible pollen, can take place, because such competition cannot take place in any other group of plants, because in lower groups of plants, whichever pollen, when the gamete comes in earlier, it will come in contact and recognition is established and fertilization takes place. And then the evolution of zygomorphy leading to floral specialization. So now most of the evolutionary biologists, they think that these are the factors which have made flowering plants very successful. Now coming to after all here, between pollen-pistil interaction, there is a continuous interaction between the haploid pollen grain and subsequently pollen tube and the diploid tissues of the pistil. So this one, the pistil, basically there are two types of styles. One is solid style and the other is hollow style. In solid style, there is a solid core of transmitting tissue connecting the stigma into the ovary chamber. Whereas in hollow style, there is a continuous cavity connecting the stigma and the ovarian chamber. Now these are let's say this is a transverse section diagrammatic sketch. You can see these are the cells of the transmitting tissue, but there is a massive intercellular space filled with intercellular substances secreted by the cells of the transmitting tissue. Now the pollen tubes always grow in these intercellular spaces. They never grow inside the cells of the transmitting tissue. So here, these intercellular spaces, they contain extracellular matrix secreted by these transmitting cells. And again, it is heterogeneous. It contains lots of components. And here, the growing pollen tubes also, they keep secreting the components from the pollen tubes. And here, the interaction takes place between the components secreted by the pollen tube and the components which are already present in these extracellular components of the pistil. And this is a longitudinal section. You can see how the pollen tubes grow in these extracellular matrix, and they don't grow inside these cells. In hollow style, of course, there is a stylar cavity, and this is bordered by one or a few layers of glandular tissue called canal cells. And these canal cells will again secrete extracellular components, and sometimes these extracellular components accumulate on the inner tangential wall, and the cuticle which was there will be pushed towards the center. And in some systems, the cuticle is digested. The whole stylar cavity, such lilium, for example, is filled with these extracellular components, and pollen tubes always grow in these extracellular components. And here they grow in this, the red ones are the transversely cut pollen tubes. So invariably, the surface of the stigma on which the pollen grain lands, there is an interaction between pollen wall proteins and the stigma surface proteins. And throughout their journey, in the form of pollen tube growth in the pistil, again, there is a continuous interaction between the haploid components and the sporophytic or diploid components. Now here, one of the things which has been demonstrated experimentally that after all, pollen grains are small. In most of the species, they are about 15 to 30 micrometers in diameter. The amount of nutrients present in the pollen grains is very limited, whereas they have to produce a pollen tube with a massive wall material, which has to grow up to 10 or 15 centimeters, like some of the Nicotianas, some of Liliums, and so on. You can see how long the style is there, but the pollen tubes have to pass through the entire length of the style. The amount of material, wall material they have to synthesize is massive. Simply, pollen grains don't contain enough material, nutrients, to produce such a huge, massive pollen tube material. So it has been shown the style provides these nutrients through radioactive label. They have been able to show that style, when once it recognizes it is the right type of pollen, then the nutrients are supplied by the pistil to the developing pollen grain so that they don't have any problem. Now, another thing is when once the pollination takes place, the pistil stigma somehow sends a message to the ovary to make everything organized so that the things are facilitated to receive the pollen tube so that fertilization can take place normally. So there are many experimental studies that have been shown. That means some sort of signal goes to the ovary to prepare the ovary to receive the pollen tube. And it also, if self-pollination takes place, the message will be different, and cross-pollination takes place, the message will be different. For example, petal senescence, that is activated even before the pollen tube reaches the ovule. Then, as you know, most of the flowers emit fragrance to attract pollinators. And when once the pollination takes place, the signal goes there to prevent or to terminate the emission of fragrance. So by the time the flower opens, development of the pollen grains and ovules are complete, but there are some in orchids. See, orchids, everything is very slow, whether the development of the flower and the pollen pistil interaction and development of the fruits and so on are very slow. Here, what happens is when once the pollination takes place, in these orchids, even ovule is not even initiated in the ovary. So this is one of such orchids. I've put it in a diagrammatic way. So at 0 week, the pollination takes place. So after 1 week, the initiation of ovule primordia takes place. And in about 2 weeks, the pollen tubes reach the ovary, but then they can't fertilize because ovules are not yet developed fully. And by the time the ovules develop, it will be 10 weeks after the pollination. Then until the 10 weeks, for example, pollen tubes, as I said, they reach the ovary 2 weeks after fertilization, and they have to wait there for 8 weeks until the ovules are fully developed. Now, what is the mechanism which makes them to cease growth and go under sort of a rest? And when once the ovules are differentiated, what makes them or what triggers them to activate themselves and then continue to go and enter the ovule and so on? These are things which we still don't know. But some of these arrangements are so fascinating. So how nicely they coordinate between the details of pollen pistil interaction. Now, the next question is, of course, I don't go into further details how the pollen tubes are guided here during the pistil. Always they grow from this end to this end. But once they reach the ovary, until here they go straight into the transponding tissue. But once they reach the ovary, they have to take a turn and get into the ovules, the pollen tubes. Now, what makes them to take a turn? All this distance they were going straight, but once they reach the ovary, they have to take a turn to enter the ovule. Now, this experiment has been very good experiments have been conducted in Arabidopsis. Now, in Arabidopsis, this is the diagrammatic structure, a stigma and a short style. And here the pollen tubes keep coming. And first pollen tube which comes, it turns towards the first ovule and enters. And the second pollen tube turns here and enters. Like this one, all the ovules will get fertilized. But there are some mutants in Arabidopsis in which some ovules have embryo sac and some ovules do not produce embryo sac. And when they studied this pollen tube growth, they found that only those ovules which have embryo sac, they receive the pollen tube, whereas those which don't have the embryo sac, they don't receive pollen tube at all. Pollen tubes start growing haphazardly in the transmitted tissue. Some of them will grow beyond the position of the ovules and so on. So what this experiment indicates is somehow the embryo sac is the one which will send some stimulus to the pollen tube so that pollen tube will turn towards the ovule and enter the embryo sac. Now, of course, initially we didn't know what is the stimulus, although they knew that embryo sac was the one which attracts these pollen tubes. But subsequently, one Japanese group, they have been doing a lot of work in one Torinia fornerae. This is one of the interesting species of Scrophulariaceae, where the embryo sac emerges outside this micropyle of the ovule. So what they did was, they made an agar medium in which these unfertilized ovules were embedded, and they carried out pollination on the stigma. And after some time, they excised the stigma and implanted this into the agar medium. Now the pollen tubes continued growth, and then they started turning and entering these embryo sacs. Now they could observe this very clearly because the embryo sac is outside this ovule. Now when they put immature embryo sacs, it didn't attract. When they put the ovules with damaged embryo sac, it didn't attract. And when they put ovules in which fertilization has already taken place, they failed to attract the pollen tubes. So it has to be an ovule in which the embryo sac is fully mature, but it is not damaged or fertilized. Now, subsequently, this is just a photograph to show this zero time, this 1.15, that means 1 minute and 15 seconds, 4 minutes and 22 seconds. This is the way how they have timed it. You can see this is the embryo sac which has emerged from the micropyle. And this pollen tube is approaching, and in this time it has reached this one. Then of course it has not entered, but it has started curving and so on. So this is just to demonstrate the entry of pollen tubes into the embryo sac, near the embryo sac. Now subsequently, they have used sophisticated experiment in which through laser beam, they could kill either egg cell or synergid cell and see what happens. So then they showed that through laser beam, they kill individual cells of the embryo sac and establish that it is the synergids which will send the stimulus. Now we know a bit about the components which attract these pollen tubes in Arabidopsis and to some extent in maize. And also there is some information about what is the receptor present in the pollen tube to recognize this, whatever is discharged from the synergids and so on. Now, of course, this is just a textbook knowledge. When once the pollen tube enters through one of this filiform apparatus to one of the synergids, then it discharges to two male gametes. One of them will enter the egg cell and fertilizes. The other will enter the central cell and fertilizes this one. This develops into primary endosperm cell, and this develops into zygote. And then, of course, you get the development of the embryo, endosperm, seed, and ovule, and so on. Now, the question is, suppose if we think that it is only the pistil which will screen the pollen grains, what will happen if you eliminate the pistil and bring the pollen grains in direct contact with the ovule? In other words, if you can carry out ovule pollination. Now, at the level of this, if you bring pollen grains directly in contact with the ovules, will the ovule still recognize that it is incompatible pollen? In self-incompatible plants, it still prevents fertilization by the self-pollen or by the cross-pollen and permits fertilization only by cross-pollen, or it accepts any, whether it is cross-pollen or other related species and so on. Now, until 1960s, there was no information at all. By that time, there was tissue culture technology was well developed, and I was a pioneer in doing this work of ovule pollination and from Delhi University Botany Department, led by Panchanan Maheshwari. This was part of my PhD work. That is, in 1960s. I joined PhD in 1964. Probably at that time, most of the participant, you people may not have even born. So what I did in Petunia is, this is Petunia, I removed the stigma, style, and ovary wall, retaining a short length of pedicel, and I inserted this pedicel into a nutrient medium in the test tube, and then brought the pollen grains in direct contact with the ovules. And then, of course, I showed pollen grains germinated on the ovule and reached the placenta, and the pollen tubes entered, and the double fertilization took place, and then all the fertilized ovules developed into seeds inside the test tube. Now, the question is whether it accepts this is self-incompatible. Petunia is self-incompatible. Now, whenever we do self-pollination, all the pollen tubes are inhibited in this style, somewhere in the middle part of this style. Now, if I put self-pollen grains here directly on the ovule, what happened? That would be the interesting thing. When I did this experiment, first, of course, I did cross-pollination, I got seed set. Then when I did self-pollination also, I got seed set. Then, of course, some statistics were done to show that there is no significance in the number of seeds that develop, whether it's self-pollinate or cross-pollinate. Now the evidences indicated that probably the ovules or embryo sac don't have the ability to recognize gametes. Whatever gametes come, they will simply accept. So I was the first to show that by ovule pollination, directly, ovules are not able to recognize the pollen grains. This is as far as self-incompatibility is concerned. So this was published in Nature in 1967. At that time, we had in our laboratory a Polish visitor from Poland who was also working in this area. So when once I standardized the technology, he was asked to do similar thing and see whether one can overcome interspecific incompatibility by using the same technique. So he did that one. When he was in our laboratory, he could get interspecific and intergeneric hybrids through this ovule pollination. And it was published earlier short note in 97. Subsequently, after he went back also, he continued to publish. Now, afterwards, of course, you could achieve this one in Poland after going back in Brassica species, Nicotiana species, and so on, indicating that it is one of the very effective techniques to overcome both intergeneric and in these self-pollination as well as interspecific pollination. The next step, of course, is in vitro fertilization. Now, as you know, in flowering plants, fertilization takes place deep inside the ovule, surrounded by it takes place in the embryo sac, surrounded by ovular tissue. Then the ovule is surrounded by ovary. So if you want to do any experimental studies, it is very, very difficult to do anything at all because of the lack of accessibility. So when compared to animals and lower groups of plants, our information about the basic aspects of fertilization in flowering plants was very, very poor. That is because of inaccessibility of the site of fertilization. So can we do in vitro fertilization as it is being done in animal systems? But when I was doing that one, we could not do that one. Then, of course, I diverted to lots of other areas and so on. But anyway, we didn't even try in vitro fertilization because there was no technique available to isolate protoplasts, isolate male gametes and female gametes and so on. Now, following progress of protoplast technology and somatic hybridization in late 1970s and 1980s, then many investigators started working on isolation of the sperms and egg cells. And so the success was achieved in three-celled pollen. So in three-celled pollen, they were able to isolate the pollen grains. As you know, in flowering plants, about 70% of the flowering plants discharge the pollen grains when it is a two-celled. That means this is the vegetative cell with the vegetative nucleus, and this is the generative cell. And in 30% approximately, this generative cell will undergo division and give rise to two male gametes. And these are three-celled pollen. This is two-celled pollen. So here the male gametes are already ready in the pollen grain. Whereas in this one, the generative cell divides and gives rise to male gametes when the pollen tube is growing through the pistil. See, this is the fluorochrome, DNA fluorochrome I have used to stain the pollen grain of two-celled pollen. This is again in Petunia, I think. This is generative cell nucleus, actually, only DNA takes fluorochrome. This is the vegetative nucleus. This is the generative nucleus. And this will divide and give rise to male gametes during its growth in the style. Here you can see this is inside the style. You can see two male gametes, and this is the vegetative nucleus, elongated one. Now, people were able to isolate male gametes from three-celled pollen. What all they had to do is you crush these pollen grains using whatever method you have, then using a sort of a density gradient centrifugation or whatever technique, then they can isolate these gametes and purify the gametes and so on. So there was no problem. First, they isolated in many species the male gametes from the pollen grains. But this two-celled pollen, they could not isolate because it happens inside the style while pollen tube is growing in the style. Now, I spent one summer in 1986 or so in Melbourne, Australia, in Bruce Knox laboratory. About 2 months I spent there. So there I took this problem. Can I manage to isolate male gametes from two-celled pollen grain? In fact, this dual system, as it was used in Torinia in 97 by Japanese worker, but I had standardized it in 1987 itself, whereas they did it by using the same technique in 1997. I did this with gladiolas. What I did was I carried out pollination on the stigma, then leave this flower intact on the plant until the pollen grains germinate on the stigma and the pollen tubes come somewhere up to here. This, of course, I had standardized the technique. Then I cut off the stigma and part of the style somewhere here and implanted it in the agar plate containing suitable nutrient medium for pollen tubes. Then the pollen tubes continued to grow and hundreds of pollen tubes emerged into the nutrient medium. And by the time they emerged, the generative cell had divided and given rise to two male gametes in all the pollen tubes. Then I treated these pollen tubes with suitable enzymes, and then I could isolate the male gametes from these pollen tubes. So these are the scanning electron micrographs of the sperm cells or male gametes I have isolated from gladiola. And subsequently, the same technique has been used. People have been able to isolate the gametes from a few other two-celled pollen systems. Now, simultaneously, the progress was achieved in isolating the female gametes. This is the embryo sac they could isolate, and any cell in the embryo sac also they could isolate. They could isolate the central cell, they could isolate the egg cell and so on. So by 1990, all the basic requirements to attempt in vitro fertilization were available. So one could isolate the male gametes, one could isolate the female gametes, they could be kept in viable condition for a couple of hours and so on. So that it was time to test whether in vitro fertilization can be achieved. So the pioneering success was achieved in Germany by one Kranz and his associates in maize. So they isolated male and female gametes, they brought them together, and then they saw that the sperm cell entered the egg and zygote was formed. This in vitro formed zygote was developed in a suitable tissue culture technology by using this nurse culture technique. And then these are the photographs of in vitro fertilization in maize. You can see this is the egg cell, this is the sperm cell. And here sperm has entered and the sperm is moving towards the nucleus, and finally it has entered the nucleus. That's using this one. Now, one thing interesting here is when the egg is intact inside the embryo sac, it is a highly polarized structure. On one side, you have the vacuum, whereas on the other side, you have the nucleus and so on. But here, you don't see that polarization. It appears to behave as a protoplasm rather than an organized egg cell. So here, of course, in protoplasm, it can easy to fuse different the cells coming from different species and so on. So here, it would be interesting because we don't have much knowledge whether it behaves like a protoplasm so that there is hardly any recognition in terms of as it happens in embryo sac, or it behaves like a protoplasm so that any male gamete that come will fuse with the egg. Now, of course, this egg was cultured, I told you, through Morse culture technique. It got the callus mass. Then from here, shoots and root were differentiated. The plant were transferred to the soil, and they show that tarsal and the carb development were perfectly normal. So this was the first report. Subsequently, 4 more reports have come up. Now, from our point of view, whether these protoplasts have the ability to recognize the male gamete and so on, they have done series of experiments. For example, maize sperm cells, they have tried to fuse with the central cell, the same embryo sac component, but they fuse because the maize sperm can fuse not only with egg cell, but it can also fuse with the central cell. But the fused products, they gave rise to callus comparable to the endosperm. Nothing else happened. Whereas when they brought maize egg with the egg of the maize itself, they fuse, but no further divisions. When the maize sperm with the egg of related species like wheat and sorghum, they fuse, undergo some divisions, but no further growth. But when they bring the sperm of maize with the egg of brassica, which are distantly related, they still fuse, but no divisions. So here, what it tells us is that this egg, under in vitro conditions, can accept practically sperm from any species, whether it is from the same species or another species, whether it is closely related or whether distantly related. That means it has no ability to recognize the male gamete. It seems to accept any approaching sperm. So in a way, concluding statement is flowering plants have evolved into a matriarchal society. So the selection of the boy, that is the pollen guys, is the prerogative of the maternal tissue or maternal parent. So the boy, their pollen grain, or the girl, the ovule, have no say in this selection. So this is what all the evidences tell us. So with this one, I conclude. Thank you very much, and you are welcome to ask the questions. I will be happy to answer. Thank you. Okay. Hello. Hello, am I audible? Dr. Khan? Hello?
Sir,
Hello?
you have lost your screen.
The avascular mycorrhiza is able to cope up the antibiotic stress. So, also populations is against antibiotic against biotic stresses. Biotic stresses particularly disease from the pathogens. It confers the antimicrobial resistance to the associated.
Thank you so much, sir, for such a wonderful. All the participants are also writing in the chat box that it was very wonderful lecture from you. Always pollination biology is the favorite concept.
Overburdened dump of soil.
Is some other lecture going on or what?
And now reforested by applying this particular avascular mycorrhizal tool. Avascular mycorrhiza are very good soil binder.
Hello?
Yes, sir. I have stopped. Actually, I was attending some other meeting also.
Okay.
I was
Now,
attending.
I mean, was my lecture audible throughout or?
Yeah, it was very much audible, sir.
Okay.
I was watching it in YouTube also. Your lecture was already live on YouTube also.
Okay. All right. Are there any questions? So
Participants, please, you can post the questions, or if not, you can raise the hand. I will pass the question to sir. All participants are writing. Mostly participants writing. It is good concept actually. Sir,
So
as we have two type of things. Sometimes there is a open kind of is there, and closed type of things are there. So how the closed one will get open, or how it will get the cavity? That is the interesting concept. Can you just glorify that concept?
No, the style from the beginning itself, for each species, it is characteristic feature. While during morphogenesis of the pistil, the cavity will be there right from the beginning in hollow style, whereas in solid style, there is a transmitting tissue, which is invariably present right from the young bud stage. So there is no the degeneration of the transmitting tissue to become hollow. If that is what you mean.
Sir, evolutionarily, when you see evolution, which forms were the earlier forms and which are the latest forms? Whether the open solid form or
most of the earlier ones had solid style.
So this is also a character for characterization.
Pardon? Hello, repeat please.
Is this also a kind of
See, the thing is, yes,
sir, is this a
as far as the style is concerned, in all the species, it has not been worked out. So nobody has checked this is the primitive character or this is the advanced character. Whatever little fragmentary information is there, that most of the primitive or earlier flowering plants seem to have solid style, and hollow style has appeared somewhere later. So as I said, people are not investigated. I have investigated only stigma region or 1,000 species, but nobody has investigated this.
Sir, one participant.
So nobody investigated.
One participant
region.
has posted a question.
Yeah.
One participant has posted a question. If pollen grains reaches to the flower stigma of other species, what will happen?
I told you they can't function unless it is a very closely related species, because they won't be having keys to open the locks present in the level of the stigma or style at different levels. So they simply don't function.
There will be no response.
No, I told you that that's what all heterospecific pollen grains will not be able to reach the ovule and achieve fertilization. So it is a waste from that point of view.
Please, participants, you can post the questions. We have some more time.
As it is my regular, this one, my PowerPoints, I have sent it to Dr. Khan. If any of you want to make use of them, you can request him and you can get those PowerPoints. This is my regular policy. Wherever I give a lecture, I make my PowerPoints accessible to the participants. So the last one, this one, and the next one I am giving tomorrow also, the PowerPoints will be available to all the participants. So you can make use of them. And if you develop any doubt or if you want any clarifications, and you can send me through email the questions or clarifications. Even later also, I will be happy to answer or clarify whatever points you raise. So my email is there on the first PowerPoint in all my presentations.
Thank you, sir. Thank you so much. Thank you. I have some announcement for the participants. Dr. Ninad. Dr. Ninad, see, I have to send the certificate. If you don't reply, I can't help because you have not given. You can unmute yourself. Dr. Ninad. Sir, if you want, you can leave, sir, meeting.
Okay. All right. Don't forget, all the best. So I will see you tomorrow, maybe around 2:15 or 2:45. Yeah. Thank you.
Dr. Ninad, you can even unmute yourself and you can start your video also. I have started video option also. Dr. Ninad, you can unmute yourself. You have given in your address some college name. I will read out. Can you reply? Can you unmute yourself and reply? Please, quickly you should respond. If you do such a delay, how can I help you? See, you have given SPM science and what it is, some arts college name. Can you speak out your college name, or can you put it in a chat box, or you send the detail address to email? You can't unmute yourself. You remove your mask and unmute yourself here. I have to speak others also. Please, Dr. Ninad, last time I am telling you, remove your mask and speak. Next, I want to speak to Dr. Rajgopal Reddy also. Sir, can you
Hello?
Sir.
Sir, please tell me, sir.
I have to send the certificate, no? So that is why I am checking your addresses once.
Yes, sir, please, sir.
1 second.
Okay.
You have given your residential address. In the residential address, you have given the flat number and Arvinda Nagar. After that, you have not given. where it is Arvindanagar?
Sir, Arvind Nagar, Kadapa, sir, Kadapa.
You have not mentioned that. Please, you reply complete details of your, if you want.
Yes, sir,
If
I
you
want.
want corrective postal address without delay, you send the correct address, because in the application online what you have submitted, no,
Yes, sir, yes, sir.
only Arvindanagar is there. So
Sir, below line I mentioned, sir, Kadapa 516001.
no, I
number
have
also
checked. I
mentioned.
have thoroughly checked it today. Everybody is, I want to say,
Okay, okay. Sir, shall I send it to my postal address to your mail ID, sir?
yes, complete details address we want actually.
Okay, sir. At present I will give immediately.
Thank you.
Only details of myself, sir.
Means your where we need to send you the certificate, that details you send me.
Okay, sir. Thank you. Sure.
Okay. Okay.
To your mail ID, sir.
Yeah, we are sending you every day the emails. No, you are not getting the email every day. Hello. Probably he lost the connection. Dr. Subbaraja, are you there? Dr. Subbaraja. Dr. Subbaraja.